There we go. And now we are. Go for it.
There we go. All right, welcome back. And real quickly, we've got a lot to cover today, but thank you to the partners. Logos and websites there for you to revisit in the recording if you'd like to check any of these out. So this is the second of three webinars, and we talked a lot about designing sheep systems last week, and now we'll delve into a deep dive in breeding and genetics, which still, if you have any familiarity with this concept, is not quite a deep dive. It's a lot to cover in 45 minutes, but we'll do the best that we can. So our description for the day, again, the highlights, covering the basics of molecular and quantitative genetics, which are going to be the very top level details, and then how to use those principles in making selection and culling decisions in a breeding program. So we'll focus on basically two different approaches, improving purebreds, and then working within commercial crossbreeding systems for the commercial industry. And you'll find that there's different concepts that apply to both as we go along. So anytime that I start with industry implications, I think sometimes it's helpful to start with natural systems, and most people are familiar with the concept of natural selection and the work of the evolutionary geneticist Charles Darwin. Most people have heard of the concept of the survival of the fittest, and the one thing that I'll challenge you to do tonight is to take that from an individual level, where you think of an individual being more fit than another, and think about this as a population. And so whether we have a natural population or an artificial farm flock population, it's still a population. And at the population level, the fittest is the population that's most adaptable to change. Now, that happens at the molecular level, at the genetic level. And the one thing that I want you to understand from tonight is that selection acts on variation, requires genetic diversity to be able to make natural or artificial selection decisions that determine which animals move forward in the population to become parents. To preserve that variation, most natural fitness traits have what's called a heterozygote advantage, which means that if the genes at a particular loci where an animal has two are different, that animal has greater fitness than if the genes are the same or homozygous. And what that does at the population level is preserve different genes so the population has the ability to adapt to change and consequently be more fit. Now that gets super deep, and we're not going to go too far into it, but I want you to think about the concept of variation and how selection acts on variation, because that's going to be important as we go through the entire evening. Okay, natural population of sheep, usually there's considerable genetic variation in wild populations. This variation is just visible in colors of soy sheep, which are relatively an undeveloped primitive breed. And again, the forces of natural selection and genetic diversity tend to preserve different genes that are responsible for different traits, in this case color, so that the population has the ability to adapt to change. Now, there's not necessarily a documented fitness advantage to one color of soy sheep versus another. But there is an example that I do want to share with you, and this goes into a different species. You can chalk it up as your random factoid for the night. But during the Industrial Revolution in England, and particularly in Birmingham and Manchester, as the Industrial Revolution increased and air pollution increased as a result, there was a species of moth called the peppered moth. Now, initially, it was kind of a grayish white color that allowed it to blend into the bark of the trees and the rocks and hide from birds and other predation. As the air quality changed and became darker, there was a gene within the peppered moth that selected or that caused the peppered moth to become darker. Okay? And that frequency increased as industrial pollution increased. And then as the air quality cleared up, the gene decreased in frequency and the peppered moths became more peppered again. Okay, just kind of a neat example. Genetic diversity, responsibility, or response to change to maintain a fitness advantage. In this case, the ability of a moth to hide from predators against a changing backdrop. But we're here to talk about sheep. So last week, we started with the concept of domestication. And we talked about how the Mouflon sheep was a descendant of all modern breeds of sheep. And through various selection for divergent products, from milk to meat to fiber, different breeds evolved from that. Basically, there was specialized selection for different purposes, which resulted in consistent types. And in the example here, the type of the merino sheep selected for wool production versus the type of the Beltex ram below selected for meat production. Okay. And that consistent type, as we talked about, was important industry applications as a big part of quality. Okay. Variation in quality is attributable to variation in genetics, variation in climate, variation in management. And I introduced last week the concept that selection is a reduction in variation. So we talk about selection tonight, again, with the goal of consistency as part of meat quality. We're going to be talking a lot about reducing variation, which is different than a natural system. Okay, but in artificial systems, our goal is to reduce variation through selection to be able to produce a more consistent, predictable product. And so in terms of controlling the variation in the genetics portion of this quality equation, we've got basically two strategies. One is selection, and the other is mating. Selection is who stays, and mating is who breeds with whom. Okay. We'll start out with selection. Selection is just a matter of moving a population in a desired direction. As we introduced last week, toward a goal or purpose of a product or products that fit into human society. So selection is a process of manipulating variation, finding out which animals stand out amongst the crowd, and whether that's a good or a bad thing. Ultimately, do those animals stay or do they go? Okay. And most traits, if the flock size is large enough, are distributed on a normal distribution or a bell curve. And if we practice selection against an extreme that we don't like, and we remove those animals from a population, over time, the population continues to be normally distributed, but it moves, in this case, from left to right in a desired direction. And the bottom line here is if you can measure the trait, you can change it. Okay. So the performance for most traits is bell-shaped curve if the flock size is big enough. Most sheep have performance near the flock average within these two dotted lines toward the center. And you've got a pretty good chance of getting the majority of your offspring close to average. They'll be on either side of average. And this is for any given trait, but most animals will be toward the center. Now, there will be some that will have high performance. Wouldn't it be nice to get all offspring that are high performers? Kind of a Lake Wobegon situation where everything's above average. Well, that's not realistic because you're also very likely to get offspring that have poorer or that are poorer performers than their parents. And this underscores a couple things. One of the main ones I want to talk about is the importance of sire selection in this situation, where a sire genetically is as much as half of the flock, and many times the performance of lambs being poorer than their female parents is attributable to a poor decision on the sire side. Okay, one thing that I want to point out is with those low performers, and I run into this situation many times, don't try to fix animals that fall into those categories or into that category. Get rid of them, remove them from the population. And one thing that I learned from an old chicken breeder that stuck with me, and I put it here with a sheep picture because it applies equally. And what you will tolerate in the breeding system is what you will have. Okay. Lambing issues, mothering issues, disposition issues, hard keepers that require a lot more feed, sheep that are parasite susceptible. If you keep them around, you will continue to have them. If you make your sheep dependent on a certain level of care, or if you allow sheep that require high levels of care, you will make them dependent on that, and their offspring will be dependent on that. So at the end of the day, there's a question of self-respect here. And do the sheep work for you, or do you work for the sheep? How does that fit into your goals? Okay, but end of the day, remember that half of every flock is below average, and I would submit that most shepherds just don't eat enough lamb. So for most of our purposes tonight, we're going to focus on those high performers and choosing those animals for replacements in the system, while also calling off of the bottom end allows us to pull the flock average up and move it or shift it in a desired direction. Okay, so this is pretty simple. All you have to do is pick the good ones, right? The problem is it's not that simple. If it was, we wouldn't be having this webinar, okay? Because what you see isn't always what you get. And if you look at this image and you see a face, lean your head to the right, put your right ear to your right shoulder, and see if you see something different. What we see in sheep is not always what we get as far as the genetics that pass to the next generation. Okay, and so to elaborate on that, we're going to do some real quick work through Animal Breeding 101, and I'm going to call it Animal Breeding 101 to 104. High-level stuff. We introduced this in the last webinar in a different context, but I'm going to put it in here today. Phenotype equals genotype plus environment. What you see is attributable to genetics of the animal plus the environment in which they were raised. Okay, and you can further break environment into climate and management. Basically, there are aspects of climate that you can control and aspects of climate that you can't control. And so from a management perspective, regardless of your religious views, sometimes what you need to do is say a serenity prayer. Grant me the serenity to accept the things I can't change, the courage to change the things I can, and the wisdom to know the difference. And that wisdom is what I'm hoping to impart a little bit of here today. Okay, most breeders breed the best of the best and hope for the best. They understand that concept. And that's good. You know, if you control what you can with management and you breed the best of the best, you're a good part of the way there toward progress. The difference is in how well a breeder is able to define the best when they're choosing their breeding animals. So that gets us to Animal Breeding 102. Phenotypes, genotype plus environment. But that genotype part breaks down into further pieces too. Okay, and this is how good breeders are able to determine or do a better job of determining which animals are better. Okay, genotype is attributable to additive genetics plus gene combination value. Okay, so at any given location, there are two genes. At any given loci, there are two alleles, if you want the genetic lingo. One comes from the sire and one comes from the dam. Arbitrarily, let's just say those two alleles have a value of 0 and 1 for allele A and allele B. Now remember, they get two of them. Additive genetics just says those values add together. And so if the animal gets an A and an A, their additive genetic value is 0. If they get a B and a B, their additive genetic value, 1 plus 1 equals 2. And if they get one of each, 0 plus 1 equals 1. Okay? Additive genetics says that A, B genotypes have an additive genetic value of 1, regardless of whether they come from a mating of AB parent or AA BB matings, or AB mated to AB animals. No matter how an AB animal is mated or produced, its additive genetic value is going to be 1. Okay, a gene combination value says that the actual genetic value is different from that additive value. And there's a lot of different situations where that can be the case. We'll talk about some of the most common, but the one thing I want you to understand is that gene combination values, unlike additive genetics, gene combination values are not passed to the next generation. Why not? Because an individual, when it becomes a parent, only contributes one of its two genes to the next generation. So the combination that comes from the two doesn't go to the next generation. So if we focus on additive, which is the part that goes to the next generation, that gives us a definition for heritability. And heritability is the expression of additive genetic variance divided by phenotypic variance, or basically what proportion of what we see is reliably passed on to the next generation. Okay. And so when we are selecting animals, our goal is to make accurate genetic decisions. And so selection is basically an estimate. When we select an animal, we're making an estimate of what its additive or genetic or heritable genetic value is, right? And so there's a lot of different ways to select that have different accuracy, which basically just means how good our estimate is. Visual appraisal is pretty common. Looking at animals, we go to sheep shows or we look at our animals in the lot and we say, this one looks better than the other, and we can make some determinations that this animal's better than another. Okay, but I'm going to submit that there are more accurate selection criteria because there are ways to get at that genetic or additive genetic value that include less noise than visual appraisal. Visual appraisal is going to include, obviously, a lot of environmental effects, a lot of gene combination effects that we can't necessarily tease out. Okay. Now, if we measure a trait in an animal, we at least have an objective measurement instead of a subjective visual. So that's a little better. We're getting closer. And if we take that measurement and we adjust it based on things such as the age of the dam, or the birth and rearing type, or the sex of a lamb as far as its adjusted weaning weight, and that's an example that you'll be able to find in the sheep industry development manual that I'll cite at the end of the page or at the end of the presentation, but that's an example of an adjusted measurement. We take things that we know are sources of variation and we account for them in the measurement so that we can make fair comparisons within a group. Okay, we can also select for a correlated trait. And sometimes this is because of sex limitations. For example, if we want to increase the age of ewe lambs at or decrease the age of ewe lambs at puberty, if we select for rams that have greater scrotal circumference, we can do that indirectly. And there are a lot of examples where sometimes it's easier to measure one trait that affects another one. And so selecting for that correlated trait allows us to make more rapid progress. Okay, next on the level of selection accuracy is progeny testing. Okay, looking at an individual's own merit and its measurements or its visual assessment is only so much, because what really matters is what that animal's offspring look like or how they perform. So progeny testing, which became very common by the 1950s, is certainly a good way to get a better estimate of an animal's breeding value than just looking at the animal itself. Okay, next, and this came along chronologically as our computing power increased, is what's called an estimated breeding value, or an EBV. This is a statistical tool we'll break into even further. And now, with our knowledge of the genome, we have genomic enhanced breeding values that can include molecular information that gives an even more direct assessment of an animal's breeding value. So, increase in selection accuracy from top to bottom. Top is least accurate, bottom is most. And it's just like a Hallmark card. Now, if you give a Valentine with a cartoon heart, certainly that's going to have more feel-good factor than the accurate description of a heart shown here in the slide. But it's not going to be as accurate. So where do you fall in the process of selection? Do you want to make selection decisions that make you feel good, such as making your decisions based on how an animal places in the show ring, or do you want to use more accurate information as an indication of the animal's genetic merit? Okay, so let's break into that EBV a little bit and just talk about what goes into that. In the purest sense, most simple sense, an EBV, or an estimated breeding value, statistical estimate of an animal's actual breeding value, is a product of the heritability for the trait times the difference in performance between that individual and the average of the contemporary group that it's raised in. Contemporary group means similar sex, similar age. And so if an individual performs a certain amount above average, that's good, but not all of that difference is attributable to genetics. The heritability allows us to determine how much of that phenotypic difference is attributable to additive genetics. Okay, and so practically, EBVs increase selection accuracy by accounting for those other parts of the equation: environmental effects, genetic effects that are additive, genetic effects that are not, and they also, an EBV for a trait includes all records on all traits for all relatives. And so there's a series of complex matrix algebra that's way behind the scenes that evaluates pedigree relationships and trait correlations for all of that information, and ultimately arrives at an estimate for an individual for a trait. That's an estimate of its breeding value. So that gets us to animal breeding 103. Phenotypes, genetic or genotype plus environment. Genotype includes additive and non-additive genetic effects. And heritability is the additive variance over the phenotypic variance. What proportion of what we see is attributable to genes that are passed from one generation to the next. The hanger here is that not all traits are created equal. Not all traits have the same heritability. Okay? And so I roughly group these traits into survival, repro, or reproductive traits, growth, and carcass traits. The survival trait heritabilities and repro trait heritabilities are relatively low. And as we move through growth and carcass, they increase in heritability. Now, to make that intuitive, I'm going to give an extreme example in terms of survival. You know, the likelihood of getting hit by a bus is not very heritable at all. Okay, but most people are familiar with the concept. If we use a muscular ram, it's going to sire muscular lambs pretty reliably. That's a difference between these traits in terms of their heritability. Okay, so, a lot of numbers here, and I want to point you to the general groups that I just described. In general, reproductive traits, probably the most important, ewe fertility and prolificacy, or the number of lambs, are roughly 5 to 10% heritable. 5 to 10% of what we see is attributable to genetics that are passed from one generation to the next. Growth traits land somewhere between 10 to 25% heritable. There's a 40 in there in an extreme case, but that's a mature weight. And carcass traits are slightly more heritable than that, roughly 25 to 45. Okay, so reproductive traits are lowly heritable, carcass traits are the most heritable. And that takes us to the next step. Okay. We have that difference in heritability for these traits, but another thing that comes into play is heterosis. Heterosis, most people are familiar with in the concept of hybrid vigor. It's the same thing. Okay. Hybrid vigor, or heterosis, is a gene combination value. It's a genetic effect, but it's not additive, which means it's not passed from one generation to the next, but it's still a genetic effect as opposed to an environmental one. The response to heterosis for these traits is opposite to their heritability. Survival traits and repro traits have high response to heterosis. If you think back to what I was talking about with natural systems and preserving heterozygosity and performance, those survival, repro, or hardiness traits are designed naturally to preserve heterozygosity, to preserve diversity. And so in most cases, a heterozygote animal that has different genes is better off from a survival or a reproductive standpoint than one that's more homozygous. Okay? And so this gets us to the application of these concepts. Selection is choosing variation and deciding which animals to move forward with the from one generation to the next, which animals become parents. When you have a highly or a moderately heritable trait, selection is an effective tool. Okay? A high percentage of what we see or what we measure is attributable to additive genetic effects. But the opposite would be true for survival or repro traits, such as twinning. And while selection for twinning is somewhat effective, it's not nearly as effective as selection for loin eye size, for example. Okay, because there are so many environmental effects that relate to whether an individual has 1 or 2 lambs, and also there are a considerable amount of gene combination value or gene combination effects that affect repro traits like twinning. Okay, so selection for a lowly heritable trait is less effective than selection for a highly heritable trait. However, remember, for each of those traits, the effect response to heterosis is opposite. So for lowly heritable traits, we have a valuable tool in heterosis. How do you get heterosis? It comes from managing inbreeding, which in many cases means crossbreeding. And also certainly managing the environment, which is a significant contributor to those environmental effects too. But we have a powerful tool for lowly heritable traits in crossbreeding for heterosis. And carcass traits in the other end of the spectrum respond well to selection, but don't have a great boost from heterosis. Now, what does heterosis mean at the genetic level? Let's look at this graphically, and hopefully it will make some sense for you. Okay, heterosis simply says that the performance of the individual is greater than the average of its parents. If we have two breeds, breed 1 and breed 2, in blue and red here, the average of those two, the additive average of the offspring, crossbred offspring, is going to be in green. Okay, and this doesn't necessarily have to be two different breeds. It can be two unrelated animals within a breed. If they're unrelated to the point that the genes come together are different, you could still have a heterosis effect. The additive average of the two parents is in green. However, many times the performance of that crossbred offspring is greater than the average of its parents. And that difference, which is highlighted here in purple, is the definition of the heterosis effect. Okay, so keep that picture in mind, and let's look at some specific traits. This is the average heterosis effect in the crossbred ewe, a list of ewe traits, and the level of heterosis in percentage. In other words, what percentage boost does that purple portion give above and beyond the green? And if you direct your attention down to the bottom of this slide, weight of lamb weaned per ewe exposed, which includes all the traits above it, or most of them, the average heterosis effect in the crossbred ewe is 18% greater than the average of its parents. Okay, the purple adds an 18% boost above and beyond the green. We'll go back to that just to see that. Okay, in the crossbred lamb, you also have heterosis as a powerful tool, and lots of traits listed at the top, but let's go to the bottom. Weight of lamb weaned per ewe exposed. Again, includes many of those different things from survival to growth to carcass traits, but an 18% boost just from the parents being crossbred. It should be becoming obvious at this point that mating decisions are as or more important to the bottom line as selection decisions in a breeding system. Okay, and I will submit that heterosis or hybrid vigor is the most powerful tool in animal breeding. And if you're trying to design a commercial crossbreeding system, heterosis is basically a free lunch. There aren't many free lunches, but heterosis or hybrid vigor, just from combining different breeds, is pretty close. Okay, so we need to talk about mating decisions, which simply is another word for breeding. Who gets to breed to whom? Okay, so let's go through some concept slides first. Deeper genetic concepts, but the goals of mating decisions are, of course, to create genetic change toward a goal which gets our domestic animal better at producing the purpose for which we've domesticated it. And if we're trying to make genetic change, there's a golden equation in animal breeding which measures the genetic improvement per year. And that's going to be an expression of 4 different things: accuracy, intensity, variation in the numerator, over generation interval in the denominator. I'm going to go through that. Accuracy is simply the relationship between the selection criteria on which we're making the choices of who stays and who goes, and the actual additive breeding value of those animals. How close is what we're using to select them to their actual breeding value that's going to pass from generation to generation. Intensity is what percentage of offspring are kept for breeding. And a lower percentage is more intense. Okay, if we only keep very few animals on the far right side of that bell curve, or the far positive side of that bell curve, that's more intense selection. By relying more heavily on elite replacement animals, we pull our bell curve in the desired direction more intensely. And variation. Variation is simply the genetic variance for a trait. How much difference is there in the population that we can select from? If there's not any genetic difference in the population, we can't move it. But if there is genetic diversity, which most times there are, we can select for desired genes and we can shift the bell curve. So those three things get multiplied by each other in the numerator, and that's all divided by the generation interval. Generation interval is simply the average age of a parent sheep when it has its first offspring. Okay, so if we think back to numerators and denominators, what are the things that increase the genetic improvement per year? Those are the things in the numerator. Increasing our accuracy, increasing our intensity, keeping fewer animals, and increasing the variation in the population. As those things increase, our genetic improvement per year increases as well. As we decrease the generation interval, as we keep turning the generations over faster and keep a younger, mature, or a younger ewe flock, for example, we increase the genetic improvement per year. Okay, some conceptual things, but this is an important, this is the key to animal breeding right here, the key equation. So if there's specific questions about this, we can return to them. But let's put it in a practical context. Okay, if it's variable and you can detect differences, you can change it. And if it's heritable, you can change it genetically. But sometimes it's worth asking the question whether that's a good idea or not. And so the questions to ask are, what traits are economically important? Are they measurable? How much does it cost to measure them? How will you keep the records and how will you use the information? And at the end of the day, is the reward for doing that greater than the cost? I've managed some large sheep operations where the feasibility of keeping very detailed records on, for example, 2,200 ewes was minimal at the time, given the facilities and given other challenges. And so I wasn't able to record on paper or in a computer program how many lambs they had or what their lambs' weaning weights were or any of those sorts of things. But I could come up with a ear notching system where I could at least identify how many lambs they had if I was there when they lambed, or if they had any problems, and I could eliminate the problems when I ran them back through the chute. So we've got to think about how that program fits into our system feasibly. And as I mentioned, this all has to fit into an economic context. So in general, survival and reproduction traits, which remember are lowly heritable, are most important to our bottom line. If animals don't survive and if they don't have multiple lambs, they tend to be less profitable in our system. We spend a lot of focus on growth and composition traits, but at the end of the day, if those traits are, or if our selection system is detrimental to survival or reproduction traits, growth and composition traits are less important. Okay. And so as we improve purebreds, again, we're breeding for a purpose. We talked about some different purposes and definitions of efficiency in the last presentation, where we're measuring outputs relative to inputs and breeding animals for desired environments. Okay, but purpose breeding is the name of the game. In commercial production, many times the best commercial purpose of a purebred is to make crossbreds. Why is that? Because what we talked about earlier with the free lunch of heterosis. That 18% boost in just being crossbred is a hard thing to come up or to compete with in terms of selection within purebreds. And so when we're making crossbreds and they're contributing to a product, at the end of the day, one of the most important things within a purebred is predictability, that they can predictably sire offspring that have the traits that we wish to add from that breed in the system. Okay. And when we're measuring or looking at predictability, a concept that becomes important is inbreeding. Okay, think about inbreeding as simply an increase in homozygosity. Inbreeding levels, percentage inbreeding, is the probability that two genes at a given locus in an individual are identical from descent. In other words, they're the same, and they come from the same common ancestor on both sides of their pedigree. Okay, as that inbreeding percentage increases, the probability that those genes are identical by descent also increases. Now, there's a lot of myth and fact and fiction about inbreeding, and we can talk about this more in the Q&A. But many times, inbreeding affects fitness traits, most noticeably, and it's going to affect them differently based on the environmental strains in the system. And so as a system becomes more stressful or more challenging, as an environment becomes more challenging, inbred animals are going to suffer a little bit more because they lack more of that heterozygote advantage for the fitness traits. And there's ways we can calculate that, those inbreeding effects, and we're not going to get into that, but I want you to understand that inbreeding is an increase in homozygosity, and it manifests in fitness traits, which remember are most economically important, and particularly as we move animals into more stressful and challenging environmental situations. Now, last time we talked about the purpose of breeds, and we said that many breeds have multiple purposes, and that's good. But just as trying to produce multiple products with the same breed can be a challenge in the system, the genes responsible for one trait are often antagonistic with genes for another. So the more traits that we're selecting for within a breed, the slower or the more challenging it's going to be to make genetic change. Okay. And again, this is why we have beef versus dairy cattle, broilers versus layers, terminal versus maternal swine, because of those genetic antagonisms. I'm not going to say that multi-purpose breeds don't have a place, but we need to recognize that that's going to affect the rate of change that we can reasonably expect. And this is probably a complicated diagram, but to boil this down, it simply says if we select for one trait, it's going to affect at least one other most times because of linkages between traits that ultimately lead, or linkages with genes that ultimately lead to correlations between traits. And so nothing happens in a vacuum, particularly when we're talking about multiple trait selection. And that's why with EPDs or EBVs, many times they're combined into a selection index that combines multiple traits or multiple EBVs for different traits that are each weighted by a relative emphasis, give us one number at the end, and they're designed for a specific animal population within a specific industry, and they're designed to keep those multiple traits in the appropriate economic balance for the industry at hand. Okay, this happens to be a desired gains index, which has a 10-year response goal for each of the traits. And this, again, gets pretty deep and complicated, but we can return to talking about selection indices more specifically if you've got questions about them. I want to spend the last few minutes talking about mating systems. Okay. And as we introduced in the last slide, breeds are classified based on trait strengths that they have, and specifically traits that they can add strength to in a system. Within the medium wool, quote unquote, meat breeds of sheep, there are some breeds, and I mentioned black faces, that tend to be more terminal, have more growth, more muscle. White face breeds that have more maternal characteristics, more mothering, more milk, higher prolificacy, lower mature size, easier care, etc. Okay, those are complementary traits in a system. Okay, where both are going to contribute economic values. And so the two concepts I want to keep in mind when it comes to designing mating systems are heterosis, hybrid vigor, again, most powerful tool in animal breeding, and breed complementarity. Are the breeds that are being combined complementary in the different traits that they offer to the system? There's lots of different systems. 2-breed rotations, 3-breed rotations, roto-terminal crosses, all designed to keep these two concepts or make use of these two concepts with varying other trade-offs. That becomes an entire other course and a topic for the question and answer session or another day. I want to close giving an example of a strategic crossing system that's used widely in the UK sheep industry, but has also been modeled in plenty of other countries and other species because it's a very elegant combination of those two concepts, heterosis and breed complementarity, harnessing those natural powers in a system that allows them to be used to full advantage. Okay, so we're going to start out with what I'm going to describe as a hill sheep. The topography in the UK is varied. The hills, marginal land, challenging environments. As you come down, and obviously this is an oversimplification in the slide, but as you come down topographically, the environment becomes less challenging. There's more feed available, more labor available, and the breeds change. The hill sheep, in this case, got a picture of a Scottish blackface, are hardy. They're tough. They don't have a great deal of production in terms of the number of lambs they produce or the milk they produce, their growth or their muscle, mostly because those are environmental costs. They're nutritional liabilities to the system. But what they can do is survive in places where not many other sheep can, and they can convert forage in that system to things that we can utilize. Hill ewes are bred to hill rams. That produces more hill lambs. They become self-replacing in that system. They're hefted to that environment. They thrive there where nothing else can. There are market lambs that are marketed off the hills for sure, but many times their value is to contribute replacements back to the system. Okay, and we're going to switch back down this hill, so follow with me. Okay, about 4 years or a little bit longer, depending on the teeth, depending on the challenge of the environment, those mature hill ewes are drafted down to lower land. Okay, and a little error in this picture, the land doesn't come with them, but they're brought down to a less challenging environment where they can have several more years of productive life. In that environment, they're bred to a crossing sire, a maternal crossing sire. Most times, that's a blue-faced Leicester. Okay, and that produces what's called a mule. In this case, a Scottish blackface crossed to a blue-faced Leicester produces a Scotch mule. There are many other varieties that just mean they come from a different hill breed. But a mule sheep, their primary purpose is to produce females. Of course, there's going to be some wethers. They go as meat lambs, but those mule lambs are intensely valuable in the system. Why? because they combine the hardiness and the instinct, toughness of the hill sheep with the added prolificacy, milking ability, size, and angularity, maternal traits from the blue-faced Leicester, and they've got heterosis. They're a crossbred animal. They've got a boost in hybrid vigor. Those ewe lambs, those mule ewe lambs, are kept as replacements in a lowland system. Okay, and so their hybrid vigor becomes maternal heterosis, and they're crossed to a terminal breed. In this case, is Suffolk. Okay, those Suffolk terminal sires or any terminal sire breed is highly selected for heritable traits of growth and carcass. All of the lambs from that cross are market lambs. Okay, and so in this system, you have a combination of maternal heterosis, the additive value from the selection of the terminal breed for carcass and growth, and the individual heterosis of the crossbred lamb. Now, in closing, is this a system that I'm recommending for Vermont? Not necessarily, because certainly we have different situation in terms of the availability of marginal land, the cost of that land, the property taxes for it, the predation risk, so on and so forth. But if you think very carefully about the system that harnesses heterosis and breed complementarity and come up with a way that a similar situation can be done here, I think that's of huge value. Okay. Now, I see a lot of crossbreeding systems that aren't really very systematic. I describe them as Heinz 57s or barnyard menageries. And if you keep crossbred animals and you maintain them in a population without design, pretty soon you get similar genes on both sides of the pedigree, and that dilutes the power of that heterosis or hybrid vigor advantage each generation. Okay? So crossbreeding systems have to be well designed to be effective. Two textbooks that I think are important here or valuable here if you want to do further reading. I've used Understand Animal Breeding in all of my breeding and genetics class. I was taught animal breeding genetics from these books. It's a little bit dated in terms of some of the molecular information, but the quantitative value of this book, I think it breaks it down. You don't have to have a genetics background to read it and understand it. Similar thing with the genetics chapters of the Sheep Production Handbook. Great reading, another great resource I'd strongly recommend. Next week, we're going to talk specifically about meat quality, but I've run out of time, and I'll take any questions that you've got.
Thanks, Joe. This is Jen again, and I just want to encourage folks, if they have questions, to put them in the chat, and we will touch on those. Actually, I have a starting question while folks are working on theirs. A starting question that I have is I've gotten this question before, and that is, in a crossbreeding system, are there concerns between working with a smaller framed breed and a larger framed breed as a terminal animal in terms of the size of the lambs, and whether that's likely to cause any lambing difficulties?
That's a great question. That's certainly a big concern, specifically with beef cattle, where the mature size of an animal has a strong correlation with the birth weight, and even in the presence of EPD selection, there's going to be some challenges with using bigger animals relative to the birth weight of a crossbred lamb. Not so much the case with sheep. I'm not saying it's not the case, but it's not as much the case. And the big reason is because sheep have multiple lambs. So more of the dystocia, more of the lambing difficulty in sheep compared to cattle comes from having lambs that are tangled, having multiple lambs as opposed to having big lambs. Okay. Now, again, it can still be a problem, but that's one of the reasons why in the three-tiered system that I showed, and I'll pull up that slide for you again. That blue-faced luster ram in the center is a very angular, long-bodied ram with a long front, a long, narrow head, and just a lot of angularity and femininity. That has a function in terms of lambing ease, because even when those mule lambs are mated to a Suffolk or a Texel, which is going to have a bigger head, bigger front end, maybe be a little more compact and meaty, the lamb is still going to have a lot of the shape coming from the blue-faced luster. So there are ways to build that into the system by design so that lambing ease is not a big problem, or not as much of a problem. But yes, if you just kind of willy-nilly take a Texel ram and breed it to a Shetland, you know, as a designed crossbreeding system, you're probably likely to create some dystocia issues from that.
You have a thank you for taking a complicated topic and breaking it down. Thank you very much for that. Curious, are there any particular crosses that you would specifically avoid, and for what reasons?
I think the biggest one to avoid is using the breeds in the wrong direction. And so, you know, the biggest thing to avoid is using terminal breeds on the maternal side and maternal breeds on the terminal side. That certainly is going to be a liability. That's working the wrong direction. As far as specific recommendations, I think there's a lot of different ways to do this. And again, as we talked last week, there's different products, different niches in the industry, and there's room for different breeds. So I'm going to steer away from the specific breed recommendations, but I'm happy to speak with anybody specifically about their individual system. And I would be glad. I'm trying to pull my my contact information back up, but feel free to reach out to me, and I'd be more than glad to talk to you specifically about your questions on breeds and your breeding system.
We have a question from Ian, who says, I'm looking to introduce Finn genetics to our Eland flock. Will this just produce tiny, unviable lambs? Currently breed 200 pure and the other 700 to Charolais belted, but we are losing prolificacy.
Okay, so this appears to be a UK request. These are breeds that we don't necessarily have as much access to here in the States, but I do have some familiarity with them, and I definitely have familiarity with Finns. Finns, and specifically Finn Dorset genetics, are a big part of the commercial sheep industry in New York, where I worked for many years, which was driven by Cornell, and specifically their STAR accelerated lambing system using Finn Dorset crosses. I had a saying when I was lambing Finn Dorset crosses that 3 wrongs don't make a right. In other words, I would prefer, in that system, I would have preferred to have 2 big, strong twins that tended more toward the Dorset side than to have 3 small lambs that didn't grow or didn't have carcass merit, or as much carcass merit, tending more to the Finn side. And so it's a matter of finding the balance in a specific trait or in a specific system. That being said, as much as we want to emphasize carcass merit and growth, at the end of the day, economic value says that fitness traits and reproductive traits are most important, with few exceptions. And so introducing Finn genetics or Romanov genetics, or in some special cases, Barula genetics or other prolificacy sources, is a great way to increase lambing rates, and by default weaning weights, or weaning rates rather. But got to be prepared that sometimes the great ones are going to be small, not viable, and not grow. And those are things that we need to select out of our system with the appropriate balance placed on the different traits.
So to follow up Ian's example, given the size of a flock like that, if you were going to try an experimental set of, if you were going to try crossbreeding, would you do that with a small percentage of the flock to see how it went? What would be your general approach to, would you just breed? Maybe this isn't just an Ian question, this is a general question. If you're looking to make some big changes across the flock, would you hit the whole flock with similar genetics, or would you divide it up into different ways?
Well, from an experimental standpoint, obviously having some different strategies to compare gives us more valuable information. That being said, most of the flocks that I deal with in this region are relatively small, and I would caution against trying too many different things and making comparisons when the sample size that you're using to make those comparisons isn't statistically viable. And so that gets into some complicated stats that we don't need to dig into, but just as a general rule, you need to make sure that you're producing enough lambs that you can make a fair comparison. So without having to make comparisons, my recommendation, if you identify a trait that you need to change to influence your productivity, make the change, but find a way to reliably make the change. And that's the biggest challenge, particularly with some of these fitness or reproductive traits. Like if we need to increase our lambing percentage, we don't know if we've reliably increased our lambing percentage until the daughters of a ram mature and have lambs of their own, and we can assess their own lambing percentage, preferably after a couple years when they've hit their production stride. At that point, it's too late to do anything about the ram if it was a wrong decision. So we need to find ways to make the most informed selection decisions for that lowly heritable trait that we possibly can. And this is where EBVs, estimated breeding values, and particularly genomic enhanced EBVs, which are relatively less available in US sheep than in other countries or in cattle. But that's where they're particularly valuable, is those lowly heritable traits that might have some sex limitation. We can't necessarily measure them in a ram until his daughters come into production. But if we pick rams with a higher number of lambs born, a number of lambs weaned EBV, we're making the most informed decision that we can about his additive genetic merit for that trait. And if you're making those kinds of informed decisions, I would recommend that you do it in a way that's significant and going to change your system, as opposed to just dipping your toes in the water.
Thank you. So Glenn is asking, what are your thoughts on accelerated breeding? How does it affect production and longevity of the ewe? Is the extra production and year-round product worth the extra cost in management?
Excellent question. And I would say that the biggest determinant of that is going to be the scale of the operation. As we talked last week, one of the biggest limitations in terms of resources in a system is labor. And the most significant time of the year when labor is important is during lambing. Lambing is the most intensive time of the year for sheep, specifically if it's during cold weather. Okay, and so if you're going to accelerate lambing by accelerating breeding, in other words, lambing on an 8-month interval as opposed to a 12, you need to recognize that that's going to mean that you have multiple lambing seasons throughout the year, and they're going to be different from year to year. So when I managed a flock in New York, I had everything on an 8-month interval. I had a group that was lambing in January and September in one year, and then May the following year. And then the third year, they would come back to May and September, and then back to January and September, and then back to the May schedule. With that system, I effectively divided the ewe flock into two parts. And so at any one of those lambing seasons, excuse me, at any one of those lambings, I had half of the ewe flock lambing in January, half of the ewe flock lambing in May, half of the ewe flock lambing in September, and same the following year, but it alternated to which group it was from time to time. That was worth it because of the scale of that operation, 2,000 ewes, and the labor that we were spreading throughout the year. And by lambing on that accelerated schedule, we were able to keep more regular lambing help. We also had the facilities to handle it. If you don't have the facilities to handle it, if you don't have the numbers to withstand the fact that sometimes lambs or ewes are going to fall out of that system and not breed, it might not be worth it. But if you're lambing on an 8-month interval instead of a 12, and you have a 150% lamb crop each lambing, that 150% becomes 225%, or 2.25 lambs per ewe per year, as opposed to 1.5. That makes a significant difference. And in terms of longevity, the maintenance period of sheep is really pretty long and pretty non-challenging. And so I think there is some room to shorten that up if you can do it in a way that you can provide adequate nutrition throughout. Sometimes that might mean that it's not on pasture using more harvested feeds or grain or whatever the case may be. But if you shorten that maintenance period up, you're actually making a more efficient system from an output standpoint. But again, you have to realize or remedy that with the inputs required in the system, specifically labor. Long answer to a short question. It could go a lot longer on that, but hope that touches the high level.
So since we are at just about 8:15, I'm gonna ask for any final questions, and I will say that we do have an additional question from Ian about in the US, is there any view on the use of mineral supplements to improve productivity, or is it all in the genes?
Well, not to be snide, but from the day or from the evening, one thing that I want everyone to understand is that nothing is all in the genes. There's going to be environmental effects to every trait, and particularly for the lowly heritable traits, those environmental effects make a more significant difference. So a survival or reproduction trait could be as much as 80 or 90% environmental and only 5 or 10% genetic. So if that's the case, there's a lot of opportunity to improve things with mineral nutrition. And we do have a lot of range in the natural mineral profiles of the soils that we've got in the US. It's a big, diverse land base. And when we're bringing animals that have been hefted to other environments, other parts of the world that have evolved on a different mineral base, sometimes we've got to supplement them differently to get them to produce the way that we want them to. So yes, this isn't a nutrition talk, but mineral nutrition is hugely important.
Cool. So it looks like we don't have any more questions in the chat box, except we're starting to get some thank yous. So I just want to say thank you so much, Joe, for presenting this evening. And I do want to invite everyone to come to the final session next Wednesday as we look at meat quality and carcass quality.
Sure thing. I appreciate everyone hanging on for the extra time, and hope it was valuable, and we'll see you next week.